Myself Prachi Saronke, and today we are going to talk about a very interesting topic which is biomolecules. So, firstly, let us understand the meaning of the term molecules. Molecule is a term which we mostly come across in chemistry. So, molecules are basically substances that are formed after interaction of atoms, and they retain the physical or chemical property of something. But in biological systems, we usually use the term called biomolecules because biomolecules are usually molecules that are produced and occur naturally. They occur naturally in living organisms. The biomolecules are organic compounds and they are very important and essential for life as they contribute in various important life processes like reproduction and also in growth and other biological processes. There are four types of biomolecules, that is lipids, nucleic acids, carbohydrates, and proteins. Let us look at the importance of these biomolecules in life processes in short. So, firstly, we have a biomolecule named as lipid. Lipid occurs in the form of phospholipids. So, as we know, our plasma membrane is a bilayer membrane. That is, it has hydrophilic head and hydrophobic tail. So, that is why it acts as an hydrophobic barrier as it keeps the cell constituents and the water differentiated from each other. Then we have fats, which occur in the form of lipids occur in the form of fats, which helps as a energy storage molecule. Then lipids also help in hormone synthesis and signaling. Then we have proteins. Proteins are one of the important biomolecules as they serve as an enzyme for various biochemical reactions occurring in the cell. Proteins also help in structural support. Now you must be wondering how proteins help in motility. Motility as in some bacteria have flagella and there is a protein called flagellin in this flagella which helps the flagella to be motile. So that is why proteins help in motility too. Then protein also help in transport and it also generate immune response. For example, we have antibodies in our system which help us to fight various diseases. The third type of biomolecule is nucleic acids. Nucleic acids are basically differentiated into two types, that is RNA and DNA. The DNA stores the genetic information of the organism and through the process of transcription the DNA replication takes place with the help of RNA. Then through the process of central dogma the proteins are also synthesized by RNA. The RNA also acts as a ribozyme and also the gene expression is controlled by this nucleic acids. Now we will look at the fourth type of biomolecule, that is carbohydrates, in detail. So let us understand what is carbohydrates. Carbohydrates are the type of organic compound which are composed of carbon, hydrogen and oxygen, and they are very crucial for uh, energy obtaining energy from various foodstuffs like fruits, vegetables, dairy products, and they serve as an energy for the cell. Then the carbohydrates are differentiated into two main types, that is simple carbohydrates and complex carbohydrates. The simple carbohydrates are further divided into two types, that is monosaccharides and disaccharides. And complex carbohydrates are divided into oligosaccharides and polysaccharides. There are various other subtypes too. Now let us understand these each type of carbohydrates in details and their subtypes too. So first type is monosaccharides. Monosaccharides are basically the most basic unit of carbohydrates. Like if we imagine a necklace, so the pearls in that necklace are the monosaccharides, which are the basic units of carbohydrates. So they have sweet taste, they are colorless and crystalline solids, and they are highly soluble in water. The most important point is these monosaccharides are called as reducing sugars because they have a free aldehydic or ketone group which can be oxidized by mildly oxidizing agents like ferric ions or cupric ions. And there are various tests like Benedict tests or Felling's solution, which helps in the detection of these reducing sugars. These monosaccharides can be further classified into aldoses and ketoses. Aldoses are monosaccharides which contain aldehyde group, for example, glyceraldehyde, and ketoses are the monosaccharides which contain ketone group, for example, dihydroxyacetone. Examples of monosaccharides include glucose, fructose, galactose. Here, if you see in the picture, the two pictures of glucose are shown, that is D-glucose and L-glucose. So your D and L means dextrorotatory and levorotatory respectively. So dextro and levo are basically formed due to the optical differences. So these two are optical isomers of each other, and that is why there are two different glucose molecules. Then we have disaccharides. Disaccharides are the molecules which are formed by joining of two monosaccharide units. And how are these monosaccharide units joined? These are joined because of the glycosidic linkages. Glycosidic linkages are formed when the water molecule is lost. So dehydration reaction helps in formation of glycosidic linkages. These glycosidic linkages can be broken down by hydrolysis reactions. Now, when we talk about disaccharides, these are divided into two types, that is, reducing sugars and non-reducing sugars. As we saw earlier, that monosaccharides, all monosaccharides are reducing sugars. But in the case of disaccharides, it is not the same. Disaccharides like maltose and lactose are reducing sugars. Now, here if we see the structure of lactose, here we don't see any free aldehyde or ketonic group, but still it is a reducing sugar because it contains a free anomeric carbon, which allows this lactose molecule to act as the reducing sugar. So, what exactly is anomeric carbon? So, anomeric carbon is a carbon in the sugar that is an aldehyde or ketone in the structure of in the open chain structure and becomes a stereo center in the cyclic form. So that is why lactose and maltose are also called reducing sugars. Examples of disaccharide include lactose, sucrose, and maltose. For example, if we see the structure of sucrose here, it is made up of two monosaccharide units, that is glucose and fructose, and the lactose is made up of two monosaccharide units, that is galactose and glucose. Then the first type of complex carbohydrate is oligosaccharides. Oligosaccharides are the carbohydrates which fall between the two types of carbohydrates, that is monosaccharide and polysaccharides. Because these oligosaccharides are made up of 2 to 10 monosaccharide units and they serve as prebiotics and they are beneficial for gut too. So the example of oligosaccharide is shown here, that is raffinose. The raffinose is made up of three monosaccharide units, that is galactose, glucose, and fructose. Now, the one of the most complex type of carbohydrate is polysaccharide. The carbohydrate is found in the form of polysaccharide inside our cells. So these polysaccharides are very complex structures and these can be branched or unbranched and they are not very readily soluble in water too. These polysaccharides can be classified into two types, that is homopolysaccharide and heteropolysaccharides. Now, when we talk about homopolysaccharides, these only contain one type of monosaccharidic units which is repeated in the polysaccharide. For example, here we have a picture of a starch molecule. In starch molecule, starch molecule is made up of two units, that is amylose and amylopectin. The only difference between amylose and amylopectin is that amylose is a linear chain, whereas amylopectin is a branched chain. That is why it is a homopolymer because there is only difference in the structure between amylose and amylopectin. That is, there is only difference between the arrangement. That is, amylose is linear and amylopectin is branched, and that is why it is an example of homopolysaccharide. Whereas in heteropolysaccharide, more than one type of monosaccharidic units are there which form a polysaccharide chain. For example, if we have an example of hyaluronic acid is basically formed by two units, that is N-acetyl glucosamine and glucuronic acid, and that is why it is an heteropolysaccharide. There are also other type of homopolysaccharide like cellulose, which contains single type of monosaccharide. So we saw about the types of carbohydrates in detail, but what is the role of these carbohydrates in metabolism? So when we consume these carbohydrates, these complex forms are broken down into various monomeric forms. Like when we eat food rich with starch, the digestion starts happening in our mouth only. So, amylase is an enzyme which acts on the starch right from our mouth, and then until it reaches the small intestine, these carbohydrates are broken down in monosaccharidic units. So, for example, if the carbohydrate is broken down into glucose, these glucose enters into the cellular respiration, and then the process of glycolysis takes place. What is the process of glycolysis? In glycolysis, the glucose is converted into pyruvate, and ATP is also generated, and that is why Glycolysis is a very important process in cellular respiration because it generates two molecules of ATP, two molecules of pyruvate, and two molecules of NADPH. And that then further, this pyruvate molecule is transported to Krebs cycle and electron transport chain. So the process of respiration is complete and all the ATP is generated and oxygen is generated. So this is what happens in aerobic respiration. But in case of anaerobic organisms or anaerobic bacteria, the pyruvate further undergoes the process of fermentation. And the fermentation is of two types, that is lactic acid fermentation and alcoholic fermentation. In such kind of organisms, the electron transport chain or Krebs cycle does not take place. The pyruvate in lactic acid fermentation is converted into lactic acid through the enzyme called lactate dehydrogenase. And this lactic acid fermentation is mostly carried out by aspergillus or lactobacillus kind of organisms. Then in alcoholic fermentation, the pyruvate is converted into ethanol and carbon dioxide with the help of the enzyme called alcohol dehydrogenase. And this alcoholic fermentation is mostly carried out by Saccharomyces kind of organisms. Now, this is all about glycolysis and aerobic and anaerobic respiration. But there is also third type of cycle which mostly occurs after glycolysis, and it also occurs in cytosol where the glycolysis takes place. This cycle is called as pentose phosphate pathway. This is a very important and unique type of cycle because here, like in glycolysis, ATP is produced, but here the ATP or NADPH molecules are not produced. Instead, the sugars that make up DNA and RNA are produced in this pentose phosphate pathway. So, pentose phosphate pathway takes place in two processes, that is oxidative phase and non-oxidative phase. In oxidative phase, the glucose 6 phosphate is converted to ribose 5 phosphate and carbon dioxide and NADPH. There is a non-oxidative phase, the interconversion of sugars of different length takes place. So this is also an important type of carbohydrate metabolism. Then carbohydrates not only help in this respiration and pentose phosphate pathway, but they also help bacteria in the formation of biofilms. Biofilms are basically microbial communities that live in a exopolysaccharide matrix. These exopolysaccharide matrix is made up of carbohydrates. And if we talk about the examples of biofilms, uh, the dental plaque which is formed on our teeth is mostly formed by the bacteria called Streptococcus mutans, and also the notorious pathogen Pseudomonas aeruginosa forms biofilms. These biofilms are mostly important for uh, bacteria as they help bacteria to be resistant against antibiotics and immune responses of the host. Then, how these carbohydrates help in cellular processes. So, energy is stored in the form of glycogen in animals and starch in plants. Also, the carbohydrates serve as an important component of glycocalyx of bacteria. For example, in gram-negative bacteria, there is a layer called lipopolysaccharide. Lipopolysaccharide is basically the combination of lipids and carbohydrates. So, it helps gram-negative bacteria to be resistant against the take-up of various stains and also against various stresses. So glycolipids also help in cell-to-cell recognition. In some pathogenic bacteria like Clostridioides difficile, glycogen is crucial for spore residence and disease relapse. Also, PHB molecules which are poly-beta hydroxybutyrate molecules are produced in bacteria under the conditions of lack of nutrients or when the carbon source is more in the medium, then poly-beta hydroxybutyrate molecules are utilized by bacteria, which are basically carbohydrates. Then the extracellular matrix is composed of interlocking meshwork and heteropolysaccharides too. Now, carbohydrates also play a role in genetic processes because the sugars that are present in the DNA and RNA, that is deoxyribose and ribose respectively, are also the form of carbohydrates. Now we'll look upon the methods to study carbohydrates. The first method is nuclear magnetic resonance spectroscopy. So this method is basically based on the magnetic field and the radiography. So in this method, what happens is the nuclei of hydrogen and carbon interact with magnetic field, and that is why these magnetic field irradiating with radio frequency pulses cause atomic nuclei to resonate along with the coupling patterns, and which produce a unique fingerprint that reveals the identity of single sugar residue. So this technique is a very effective technique as it provides structural elucidation. It helps us in determining the structure of monomers, oligosaccharides, or polysaccharide or glycoconjugates. And it also carries out quantitative and qualitative analysis of carbohydrates, probes molecular interaction, and it can be used in solid and solution states too. Now there are also some limitations of this method called NMR. So what happens is the absolute configuration and polymerization cannot be determined by NMR only. So the other methods should be used. And also these method can hardly be automized. That is, uh, only humans can operate this method for now. The second method is HPLC, which is high performance liquid chromatography. So this method is basically based on the chromatographic technique. So in this method, the two types of phases are present, that is mobile phase and stationary phase. The carbohydrate sample is like released in the mobile phase, and then according to its affinity with the stationary phase, it interacts at different levels with the stationary phase, and then we can detect this carbohydrate using detectors like refractive index or evaporative light scattering detectors like the method called ELSD. So these methods can be used to detect the carbohydrates, and this method can also be a very effective method for studying of carbohydrates. The advantages of this method that is that it provides high sensitivity and high selectivity, and it also provides multiple selection options, that is detection option, that is refractive index and ELSD. And these detection options also are non-destructive, and hence it does not interfere for further analysis of carbohydrates. There are also some disadvantages of HPLC method, that is it excels at separation and quantification, and while it does not directly provide detailed structural features like linkage types or anomeric configuration on its own, its efficiency in preparing a pure sample for other methods or for other quantification is very high. So we can conclude that NMR method is more effective than HPLC method. So this was all about carbohydrates. Thank